Hello dear students, today we will study about inhibitors and uncouplers of electron transport chain and oxidative phosphorylation. So first of all we will see the inhibitors of electron transport chain. These are the inhibitors that arrest respiration by combining with members of respiratory chain rather than with the enzyme that may be involved in coupling respiration with ATP synthesis. Okay, so the various inhibitors of this category we will discuss over here. First one is rotenone. Rotenone is a compound that is extracted from the roots of tropical plants. It complexes with NADH dehydrogenase and act between the iron sulfur protein and ubiquinone. Rotenone is relatively non-toxic to the fishes and insects as it readily passes into their gills and breathing tubes respectively. You can see here the structure of rotenone. Next, pyridine A. It is an antibiotic that is produced by a species of streptomyces, and it is a structural analog of ubiquinone, that is the electron transporter in the electron transport chain, and it resembles their structure, and it prevents electron transfer from iron sulfur protein to ubiquinone. Then the third one is amityl, that is also known as amobarbital. This also block NADH dehydrogenase complex, that is the complex 1, and prevent transfer of electrons from iron sulfur protein to ubiquinone. Next one is this is the structure of pyridine. Then the next one is antibiotic antimycin A. This antibiotic is produced by streptomyces. They inhibit the respiratory chain at or around complex 3 and block electron flow between the cytochrome B and C1. This block can be bypassed by the addition of ascorbate, which directly reduces cytochrome C. So any inhibition with this antibiotic antimycin A can be bypassed by addition of vitamin C or ascorbate. Okay, so this is the structure of antibiotic antimycin A. Next one, cyanides. All of you know that cyanides, they enter the tissues very rapidly so that a sufficient quantity becomes lethal within a few minutes. Maybe you heard about it. Within a few minutes, it's a person get died. Okay, so the cyanide ion actually combines tightly with cytochrome oxidase enzyme, leading to the cessation of transfer of electrons to terminal oxygen. Then the next one inhibitor is azide. It blocks the electron flow between the cytochrome oxidase and oxygen. Azide reacts with the ferric ion of this carrier. And the last one inhibitor of ETC is that is carbon monoxide. It also attacks between cytochrome oxidase and oxygen, that means the terminal transfer of electrons. But unlike azide, carbon monoxide inhibits the ferrous form of the electron carrier, and azide react with the ferric form of the carrier. Okay, now the inhibitors of oxidative phosphorylation. Actually, oxidative phosphorylation, that is coupled to the oxidation of substrates. Okay, so this actually this compounds inhibit electron transport in cells in which such transport is coupled with ATP synthesis. Okay, so the first one is example of inhibitors of oxidative phosphorylation. The first one is oligomycin. These polypeptide or antibiotics are obtained from various species of streptomyces. They inhibit the transfer of high energy phosphate to ADP and therefore also inhibit electron transfer coupled to phosphorylation. All of you know that the FO component of ATP synthase we have already seen in oxidative phosphorylation, in the video of oxidative phosphorylation. Okay, so the FO component of this ATP synthase enzyme actually binds oligomycin, which is a potent inhibitor of this enzyme and of oxidative phosphorylation. The next one is DCCD. The full form is dicyclohexyl carbonamide. Again, it blocks proton flow through FO component of ATP synthase. Then the next one is atractyloside. This toxic glycoside is extracted from the rhizomes of Atractylus gumifera, that is a plant native of Italy, and it blocks oxidative phosphorylation by competing with ATP and ADP for a site on the ADP-ATP antipode of the inner mitochondrial membrane, and hence it checks renewal of ATP supply in the cytosol. That means ADP cannot enter, so it cannot be converted into ATP, and evidently acetylate inhibits at a step beyond that, blocked by oligomycin, the next level. Okay, next one. Uncouplers of oxidative phosphorylation. There is a difference between inhibitors and uncouplers. Inhibitors only inhibit the synthesis of ATP molecules, but uncouplers actually uncouple the oxidation or electron transportation with phosphorylation of ADP. Okay, so uncoupling agents are compounds which dissociate or uncouple the synthesis of ATP from the transport of electrons through the cytochrome system. This means that the electron transport continues to function, leading to oxygen consumption. But phosphorylation of ADP is inhibited in the presence of uncouplers. The free energy released by electron transport appears as heat rather than a newly made ATP, and an uncoupling agent greatly enhances the permeability of the inner mitochondrial membrane to protons. They are hydrophobic weak acids. That's why they increase the permeability of protons. Their hydrophobicity allows them to diffuse rapidly across mitochondrial membrane and enters into the mitochondrial matrix in the protonated form. And then they release a proton, thus dissipating the proton gradient. Actually, they collapse the proton gradient by entering this proton, or by releasing this proton into inner mitochondrial matrix. So certain chemical compounds also cause uncoupling without disrupting mitochondrial structure. The one of the example of uncoupler is 2,4-dinitrophenol. The uncoupling action of 2,4-dinitrophenol is due to the fact that both the phenol and the corresponding phenolate ion are significantly soluble in the lipid core of the inner mitochondrial membrane. That means they can easily pass through inner mitochondrial membrane. The phenol diffuses through the core toward the matrix, where it loses a proton, and the phenolate ion then diffuses back toward the cytosol side, where it picks up a proton to repeat the process. You can see here in the structure, in the cytosol, phenolate ion accept a proton and converted into 2,4-dinitrophenol form. Then 2,4-dinitrophenol form enters through mitochondrial membrane into mitochondrial matrix. Then 2,4-dinitrophenol then releases proton and converted into phenolate ion. That means whatever the proton gradient formed due to electron transport due to this 2,4-dinitrophenol, it collapsed. Okay, that means there is no proton gradient formation, and actually protons are not entering through ATP synthase. So the energy that is released due to collapse of proton gradient released or dissipate in the form of heat. They not utilized to synthesize the ATP molecules from ADP. Okay. Now, next one example of uncoupler is dicumarol. Dicumarol arises from the action of microorganisms on coumarin. What is coumarin? Coumarin is a natural constituent of sweet clover. It is an action identical to that of 2,4-DNP, and dicumarol is also an antagonist of vitamin K function. Then the next one uncoupler is triple CP, that is full form of this is carbonyl cyanide chlorophenyl hydrazone. Its action is also similar to that of 2,4-dinitrophenol. That means outside the mitochondrial matrix, they pick up a proton and converted into protonated form, then diffuses within the inner mitochondrial membrane and enters into mitochondrial matrix, and then again back release the proton. So there is no proton gradient formation across the inner mitochondrial membrane. Next, ionophores. This is the next category of uncouplers or inhibitors, that is ionophores. Actually, ionophores, that is also known as ion carriers, are lipophilic substances. They are capable of binding and carrying specific cations through the biological membranes. In case of uncouplers, they actually transfer protons, but in case of ionophore, they differ from the uncouplers in that they promote the transport of cations other than proton through the membrane. That is the main difference. Ionophores uncouple electron transfer from oxidative phosphorylation by creating electrical short circuits across the mitochondrial membrane. These agents bind to inorganic ions and surround them with hydrophobic moieties. The ionophore metal ion complexes pass and easily they can pass easily through membranes. Example of one of the ionophore example is valinomycin. The toxic ionophore valinomycin form a lipid soluble complex with potassium ion which is abundant in the cytosol. Whereas potassium ion penetrates the mitochondrial inner membrane only very slowly, the potassium valinomycin complex readily passes through, and the influx of positive ions neutralizes the excess of negative charges inside the matrix, and it diminishes the electrical component of proton motive force. And valinomycin slows mitochondrial ATP synthesis without blocking electron transfer to oxygen. Okay, so it is just a type of short circuit. Then the last one, that is uncoupling protein. Okay, till now we have studied the uncouplers, that are the chemical compounds. Okay, but here this is the uncoupling protein, that is known as thermogenin. Actually, in most of mammals, including humans, newborns have a type of adipose tissue that is called brown fat, in which fuel oxidation serves not to produce ATP but to generate heat to keep the newborn baby warm. This specialized adipose tissue located at the back of the neck of human infants, and it is a brown because of the presence of a large number of mitochondria in it. And the mitochondria of brown fat are like those of other mammalian cells in all respects, except that they have a unique protein in their inner membrane that is known as thermogenin, and it is also called the uncoupling protein. And this thermogenin protein provides a path for the protons to return to the matrix. That means protons without passing through the ATP synthase complex, and as a result of this short circuiting of protons occur, and the energy of oxidation of substrate conserved by ATP formation, but here it is dissipated as heat, and it contributes to maintain the body temperature. So these are the inhibitors of electron transport chain, then inhibitors of oxidative phosphorylation, and uncouplers of oxidative phosphorylation.